Welcome to the Center for Misfolding Diseases, or the CMD. We're here in the Chemistry of Health building, which was specifically built for us a few years ago. So we have really excellent facilities which enable us to do really exciting world-class research. So what we actually study here is neurodegenerative diseases primarily, or protein misfolding diseases, in which proteins misfold and aggregate in the brain, resulting in the formation of amyloid fibrils. And actually this process of aggregation causes toxicity, and that's actually what causes the diseases such as Alzheimer's and Parkinson's. So what we try and study here in the Vendruscolo group is two things. We try and understand the mechanism of how this aggregation happens, but we also try and understand how we can treat or prevent this aggregation. So we do things like small molecule or antibody design that can specifically target these proteins to prevent them from aggregating and misfolding, thereby preventing the disease. And so ultimately what we're trying to do here is to develop treatments for these neurodegenerative diseases. So now that you guys know what we do here, how about I show you the lab? Okay, so here we are in front of the plate readers. This is probably one of the most heavily used piece of equipment in the lab, and as you can see we have 5 of them, which is really good because it means we can do a lot of simultaneous experiments. And what we usually use these for is to look at aggregation of protein, and so we can see how the protein assembles into amyloid fibrils. And this is also really helpful because we can look at things like drug screening and see how small molecules and antibodies affect the aggregation kinetics of the proteins. So now that we've seen those, we can move on to some of the other pieces of equipment which are over here. So here we have 2 other pieces of equipment, an FTIR and a CD, and we use these to characterize the chemical properties of our proteins or samples, so we can look at things like secondary structure or how the protein is folded. And this is really helpful that we have these here, and not a lot of labs will be able to have these really heavily used pieces of equipment here. So it makes it a lot easier to do our experiments and actually look at our sample and how it's changing. And with that, we can actually move on to the main lab and look at the equipment down there. So here we are in the main lab, and this is where we do most of our prep work and a lot of our experiments as well. So we each have our own bay here, which means that we have plenty of space to actually work and do our experiments. So here we are in the desk area, so we can sit here and work when we're not in the lab and doing things like data analysis. So it's really nice to have a dedicated space in the lab to be able to do that. Okay, so here we are in the basement of the Chemistry of Health building, where we have dedicated facilities specifically for producing protein. So obviously that's really important because we study protein, so we make all of our protein down here. And as you can see, we have a lot of equipment which is specifically tailored for this, which enables us to actually do our research and produce as much protein as we need to be able to do all of our experiments.